So here I'm going to sterilize these media samples. This is a nutrient broth. It's going to go into this pressure cooker. And the pressure will allow the temperature to reach above the boiling point of water, about 120 degrees Celsius. And that will kill any microorganisms or spores that happen to be in these flasks. I now have the heat on. This valve is closed, so pressure is building up inside. This is a pressure gauge here that is also related to temperature. I'm going to cook this in the green zone here for about 20 minutes. So now it has heated for the requisite 20 minutes. I'm going to release the pressure.
Okay, vortex it?
The pressure is being released. You can see the pressure dropping on the gauge to the left. It now smells of luria broth, which most people think is disgusting. This is now sterile media. There are no microorganisms in there, and it should remain sterile until we remove the lids, the aluminum foil lids.
That's what I'm doing.
You will intentionally inoculate this broth with bacteria so that we can grow up a large number of them, of a specific strain, of a specific, if you will, race of bacteria.
We don't have to draw the ions.
This is a plate in a petri dish. Each one of these whitish circles represents a bacterial colony. So it started as one cell and it has grown up into millions. And this plate has ampicillin, which kills most bacteria. So the only bacteria that can grow here are those that contain a gene for the enzyme that destroys ampicillin. And this enzyme is what we want to isolate. So these are a good source for that enzyme. I'm going to do what's called pick a colony. I'm going to use a sterile toothpick and touch one of these colonies and throw that into the media, the liquid media that I've sterilized. And then that will grow up into billions of bacteria in the liquid culture. So I have grown what's called an overnight. I started with sterile media, and into that sterile media I took 1 toothpick's worth of bacteria off the plate that I showed you and set that in an incubator overnight, kept it about 37 degrees Celsius. That's the same as human body temperature. These are E. coli. They're adapted to growing at that temperature. So the haze here is due to the cells that have grown in the media. I will now refrigerate that over the weekend, and you will use that. You'll use this culture to inoculate your broth on Monday. So on Monday, your goal is to take a sample of broth that you've been given. This is 50 milliliters of LB, luria broth. You want to grow bacteria in here. We will want to be able to stir this so that the bacteria get enough agitation to grow well. So a stir bar will be required. However, the stir bar right now has probably lots of microorganisms on its surface. We'll want to sterilize the stir bar prior to adding it to the bacterial broth. We'll do that with 70% ethanol. 70% ethanol is very good at killing bacteria. So once we dip the stir bar in there, the bacteria will die. However, we don't want to add ethanol to the broth because that may inhibit the growth of the bacteria. Then, once we have the stir bar in there, we'll add some of the bacterial culture, the overnight culture. Recommended quantity is 250 microliters of this overnight culture added to your sterile broth. We'll also need to add a selecting agent. We want only bacteria that are resistant to ampicillin to grow in the flask, so we will add ampicillin. It will kill bacteria that do not possess the enzyme we desire. Only those bacteria that can break down or destroy the ampicillin will survive. So we'll add 500 microliters of that. So I'm demonstrating how to sterilize a stir bar. I have a nice clean set of forceps here. Don't use rusty old forceps. And I grip the stir bar securely, and I dunk it in 70% ethanol. Hold it there for a few seconds. It is now sterile. I will now flame off the residual alcohol. This flame is not very hot. I just bring the stir bar there. The ethanol is burning. It's not a very hot flame. It's not hot enough to melt the Teflon. This stir bar is ready to go plunk into the flask. So I would lift up the lid and plunk it in. In this biochemistry project, there will be times when you have waste that is actually a biohazard. We do not want these bacteria, which are resistant to ampicillin, to get out into the environment. So, if you have things that have touched the bacterial culture, for example, like this pipette tip, we will dispose of them in the biohazard waste vial or jar. Each group will have one of these. I just inject the entire pipette tip into the biohazard waste, put the lid on, swirl it up a little bit. This will just collect your biohazard waste throughout the project, and we'll clean it out at the end. I have here a stock solution of 10% bleach. That's what goes in the disinfecting chamber, and that should be enough for the whole class.